Hello everyone, welcome to the Notes channel. Today I am here with a very known topic of a plant biotechnology is a protoplast culture. Now, before going for a culturing process, we will understand what is the protoplast. Protoplast is, first of all, it was discovered by Hansen in 1880. Now, and this is nothing but a naked plant cell without the cell wall, but they have a cellular component and they possess a plasma membrane. This is nothing but a naked plant cell without the cell wall, but they have cellular component and they possess the plasma membrane. Basically, the protoplast denotes the functional plant cell, but for a lack of a barrier cell wall. This protoplast, they don't have this cell wall along with them, but they possess the cellular component and plasma membrane. Now we will see how this protoplast is going to isolate it from the cell. It is isolation of this protoplast can be done by mechanical method or enzymatic method. Here, this mechanical method is mostly it's not preferred. It's just because of the yielding by a mechanical method is very low, and this procedure is very crude and tedious procedure. I'll write it here. Mechanical method first of all, it's a very crude and tedious method, and it's produce a very low yield. So the most of the workers, they didn't prefer this method. They went for a enzymatic method. This mechanical method following some stages for the isolation of a protoplast, like small pieces of epidermis from a plant is selected. And then these cells are subjected to a plasmolysis by keeping the cell in a 0.1 m sucrose. Keeping the cells in sucrose, 0.1 m sucrose, it went for a plasmolysis. Keeping the cell in a 0.1 m sucrose cause a plasmolysis, and that cause cell to shrink away from the cell wall. As soon as the cells shrink away from the cell wall, the tissue is dissected. As soon as the cell to shrink away from the cell wall, the tissue is dissected to release the protoplast. So basically, this is the method where we are going to use the epidermis from a plant and keeping the cell to a 0.1 m sucrose that will cause a plasmolysis. And this plasmolysis helps to the cell to shrink away from the cell wall. Once the cell wall shrink away from the cell wall, the tissue is dissected and the protoplast is isolated. The mechanical method for the protoplast isolation, as I said, it's very low yielding, but low yielding, and their viability is also low. and this method is restricted for a tissue, certain tissue with the vacuolated cells. So it's not no used for a vacuolated cells. And this is, I already said, it's a crude and tedious method. So this is the procedure we are going to use to isolate the protoplast from a leaf or any cells, that from any cells, like small piece of epidermis for the plant, cell in a 0.1 sucrose that cause a plasmolysis, cell to shrink from the cell wall, and tissue is dissected to release the protoplast. Now we will talk about the enzymatic method. Enzymatic method is the most common method used for the isolation of protoplast by using the enzymes, and this method is mostly preferred by the worker. Now we will see the enzymatic method, which is the most widely used or most preferred method, just because it produces a high yield of viable cells and the minimal or no damage to the protoplast Here in this method we are going to use the enzymes for isolation of protoplast from the various wide variety of tissue or organ, like leaves, shoot apexes, fruit, embryo, etc. So this enzymatic method is done the procedure of enzymatic is of two types. First is a direct method, means direct or means two step. One is the two-step method, or second is a one-step method. In two-step method, mostly it is called as a sequential method, and this is called as a simultaneous method. This is sequential method, and this is simultaneous method. Here, by pronunciating the sequential method and simultaneous method, we can understand that the two-step method, in that the tissue or a cell is going to treated with a first with a macroenzyme, as we can take an example of pectinase, or second with a this is giving this macroenzyme is just giving to degrade the middle lamella of a tissue, and then it exposed to the this microenzyme just to release the protoplast. As we take the example of a leaf, we are going to take a microenzyme as a pectinase that will degrade the lamella, and the microenzyme as a cellulose. But in the case of a simultaneous method, it's a one-step method. Here, the protoplast isolation done by involving both macro plus micro enzyme simultaneously. Now, while pro- isolating the protoplast from enzymatic method, at the time the uh, pH should every time the pH should be 4.5 to 6. The pH should be maintained 4.5 to 6, and the temperature should be at 25 degrees to 30 degrees Celsius. 25 to 30 degrees Celsius. Enzymatic method, the most preferred method, just because it's yield a high viable cells and the minimal where the no damage of a protoplast. The isolation of protoplast should be done at the pH of 4.5 to 6 or a temperature 25 to 30 degrees Celsius. Enzymatic method can be done by two different way. First is a 2-step or second is a 1-step. In 2-step, we are going to take two different enzyme at a different time. But in a 1-step, we are going to take both the enzymes at the same time. So that will cause the release of a protoplast. That's the two method we are going to use for a isolation of a protoplast. Isolation of a protoplast from the tissues, leaves, fruits, and embryo. Now the application of protoplast. The application of protoplast means how we can use this protoplast in the different kind of cultures and all. So it can be used in the production of cybrid, means the cybrid where we are going to fuse the two different kind of a protoplast, and in that the new offspring having a one nucleus from any one of the parent and the cytoplasm from the both of the parent, and this can also be used for a somatic hybridization, just because to overcome the sexually incompatible species. Here, this also can be used for ingesting foreign material into the cytoplasm. This application of protoplast can be also done for a DNA transformation or study as a single cell system, along with the used to study a wall synthesis and the decomposition. Basically, the protoplast is very useful for every process. If you like the video, please share and subscribe. Thank you.